Hello, everyone. In this chapter, we're going to talk about viruses. So, we're going to talk about why we study them, some of the traits we use to study them, some of their diversification events, and some of the key lineages using the Baltimore system. A virus is an obligate intracellular parasite. It has to go into a host in order to use its machinery to reproduce, and every little particle of the virus that's infectious is known as a virion. They typically don't have all the characteristics of living things. Um, For example, in terms of hereditary material, they do have DNA or RNA. It could be single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA. It doesn't have a plasma membrane, or they don't have plasma membranes, but they do have an envelope. They don't do their own transcription. However, they do have some enzymes that do help them in this process once they infect a cell. They cannot carry translation independently. They do need machinery for that from your cells, and they typically have zero metabolic capabilities. So, why do we study viruses? Viruses do contribute a lot to organismal evolution. They are one of the things that is still selected for, especially things like good immune systems in current populations. They promote mainly anything that has to do with some type of defense mechanism for your body to prevent infection. In this case, we have our immune systems. Not only that, but they're also really important for lateral gene transfer. They can take pieces of DNA from one organism that they've infected and take it into another one. In fact, about 5 to 8% of the human genome is remnants of these viral genomes. One of those actually responsible for the development and the function of the placenta. So the reason we give live birth is because we have or we had that viral infection. If you think about everything else on the planet, everything else lays eggs, but we have live birth. So the viruses affect just about every system in our body, and this chart kind of shows you the different types of viruses and where they infect. An epidemic is typically a very rapidly increasing disease in terms of numbers of infected cases in people. Um, a pandemic is a worldwide spread. So the Spanish flu, for example, of 1918 to 1919 killed up to 50 million people worldwide. We also see the same thing with the coronavirus. It's now worldwide and it's going up in cases every day. So again, here we see that population of people, and again, there have been different viruses through um, a lot of different periods of time, and here we see that dip in life expectancy. Again, close to the 1920s, you start having people that were dying, not even making it to their 40s. HIV, or the human immunodeficiency virus, causes AIDS, or acquired immune deficiency syndrome, and it's affected more people than measles, smallpox, and influenza worldwide. It has already killed 37 million people worldwide, and there's an estimated 37 million people infected um, currently. One of the most hard hit places was the sub Saharan African region, with 29% of the um, Swaziland population being HIV positive. It is a sexually transmitted virus, so it disproportionately affects young adults. There's no cure. There are treatments, however, they delay the onset of AIDS. Um, so without treatment, those that have HIV in their late teens or 20s usually would die by the age of 30. What HIV does is it basically goes and infects your helper T cells, which are the cells that help mount an immune response, and it injects its genome into them. Then, as those cells replicate, because again, let's say you have an infection and you need more white blood cells, your body triggers the production of more of them. So whenever these are replicating, they're basically copying themselves with a virus inside them. So as they continue to replicate, they grow in numbers, and then at some point they wake up from their dormant stage and they start creating new virions, and then all um, all of those little virions are going out and infecting other cells, eventually killing them, disabling your immune system. So your body cannot keep up with the production um, of those white blood cells. So you do have an acute phase. So typically, the first 6 weeks or so, the virus is kind of replicating, and then um, after a certain point, I think it's after you have less than 200 helper T cells per cubic millimeter of blood, then it develops into AIDS. So at this point, any infection could be lethal. Bacteriophages usually infect bacterial cells, and they have actually been used in the past to study some of the processes that viruses use in order to infect and take over the cells. But we've actually gotten a lot of interesting things from them. The vaccines are basically inactivated forms of the virus that the immune system learns to deal with. So whenever you inject those little particles in you, your immune system picks them up, it attacks them and destroys them. And one of the things that's left over from those infections are memory cells or B memory cells. And whenever your body comes across that, let's say that it exposes these little proteins, once the body recognizes those little proteins again, then it basically tells the white blood cells, hey, it's this infection again, let's go after it. So it starts replicating those cells that can kill that pathogen, and then they go after it. Another thing that's come out of that is the antiviral drugs as well. So they are any drug that interferes with the replication of the virus. Virions are typically anywhere between 20 to 300 nanometers in diameter, and compared to a eukaryotic cell or even bacterial cells, they're really tiny. And again, Koch's postulates are really important for establishing the cause of the disease. Once the virus is isolated, you can get the structure, the nature of the genetic material, and the mechanism by which it replicates. These are all very important and essential for trying to develop either treatment or a vaccination for this virus. Here we see the comparison between the HIV virus, a bacterial E. coli cell, and a eukaryotic red blood cell. When it comes to the morphological traits, these viruses are quite simple. They typically all have a protein shell that's called a capsid, and then they have some type of protein or something on the surface. Some of them have what's known as an envelope, which is kind of like a membrane-like layer. The capsid helps protect and release the genome, and the membrane helps it attach. So here you can see different viruses. You have the TMV, which is the tobacco mosaic virus, the adenovirus, the bacteriophage. So I think most people are familiar with this one because of like Hollywood and the way that the media portrays viruses. Those only affect bacteria. They don't affect any of your cells, for example. And then you have enveloped viruses, which are the ones that pretend to be your cells, but they're really not. When it comes to the nature of the genetic material of a lot of viruses, there is a lot of diversity. So I'm sure you guys remember the central dogma, which is DNA makes RNA, and then RNA makes proteins. However, the genetic material of viruses actually sometimes starts with RNA, and then goes to DNA, and then goes to RNA again, and then it goes to proteins. Or sometimes it starts with a single-stranded DNA, and then it goes to a double-stranded, and then it goes to RNA, and then it goes to protein. So the way these viruses replicate and the way they make their proteins is not consistent with the central dogma of molecular biology. They kind of do their own thing. So you have positive sense genomes, which are genomes that have the same sequences as the mRNA that you need to start producing proteins. So the positive sense sections of viruses, if it has that one, sometimes it has a negative sense, but if it has a positive sense genome, it can start making some of those proteins right away. The negative sense genomes basically have the complementary strand to the positive sense. So once that positive sense strand is made, then that can go and start making proteins. Some are ambisense, so they have both the positive and negative, so they can be both making templates and also making proteins at the same time. There are two patterns of growth. You have the replicative growth, which is typically the one that quickly makes a lot of virions, and usually ends in the death of the cell through lysis. And then you have the dormant manner or the lysogenic manner. So this one is when the virion production is suspended because the DNA is basically getting combined with your DNA and blending so that the cell doesn't notice that it has anything different. So the virus and the host coexist for some time. Every new cell that is made from that cell that it's infected contains a copy of that genetic material within its DNA. So that's why it's so dangerous. Your body and your immune system aren't able to detect it because it's now part of your cells. There are 6 phases to this replicative growth in all viruses. So the first is the attachment and the entry to the cytosol. Then this genome that the virus has gets transcripted um, and then it gets translated. Then you have the replication of the genome or the genetic material. Then the new virions assemble, they exit, and then they transmit it to another cell. That lytic cycle, that's the one that ends with the typically the rupture of the cell, and one virus can make hundreds of other viruses. So again, here we have that little virion infecting that cell, um, and then you have the DNA making RNA and then proteins if it's a positive sense, and then it goes starting to manufacture the parts, then it becomes assembled, and then they get released. Typically, this type of growth has like a stepladder pattern to it, where the division of the cells may be linear. However, the um, production of these phages in this case is exponential almost. So how does the virus enter the cell? First, it has to become attached to the cell, and then it has to go into the cell, and then it becomes uncoated. And this uncoating usually is what dissolves the membrane or the protein capsid so that the genome can be exposed and gets to work. In the case of HIV, the HIV enters the cell only if there is a protein called CD4. So your immune cells have a CD4 receptor. So this CD4 receptor identifies that virus as, from the body, so it takes it in. However, um, what it doesn't realize is that it's a virus. So that cell becomes infected. Some of the trials and some of the medications and the treatments that are available for HIV can sometimes block that CD4 receptor. The problem with blocking the CD4 receptor is if you block the virus from entering, you also block any other signals from getting through to those immune cells. So mounting an immune response is going to be difficult. There's also in this case a co-receptor, which is the CD8 receptor, which is kind of like a double checkpoint system to make sure that it's actually binding to what it needs to bind. Um, so this HIV virus is actually able to bind to that as well. So it really tricks the cell into thinking that it's coming from your body. So again, the HIV virus binds to the CD4 receptor first, and then to the CD8 receptor, which is right next to it, and then it's able to fuse with the membrane and then enters your cells. And then in there it becomes encoded, and then it starts making copies or embedding itself into your DNA. Um, there's also the process of endocytosis in which basically it pulls the virus into a vesicle, and then once it's inside, it kind of takes that membrane off and it ejects the contents um, into your cells. This is an example of the influenza virus. The influenza has maybe, I think, 6 or 7 little parts to it, or DNA sequences to it, and then once it makes its way into your cell, sometimes it recombines with other strains that are also there. Antivirals are any type of drug that interferes with the infection or the replication of the virus. Because viruses don't have ribosomes, amino acids, ATP, or anything biosynthetic in nature, they require your or the host's machinery to work. Um, so these uh, viral mRNAs end up getting translated in one of 2 ways. First is through the ribosome on the rough endoplasmic reticulum, and those that are non-enveloped typically use the ribosomes that are free in the cytosol. In the case of HIV, there is an enzyme that is actually really important for the maturation of the virus, and that is protease. So what the protease inhibitors do, which is a type of drug, is they go into the cell and they block those protease, so that protease activity is disabled. So whenever this is disabled, it interferes with the replication of the virus, either by not allowing it to replicate its genome or not allowing the virus to assemble so that it cannot infect anything else. Here we have the active site of protease, and over here you have the competitive inhibitor that basically blocks the site, making it unavailable. So how do they copy their genome? It really depends on the type of virus that you're looking at, but overall they all have the same thing. They want to make sure that they are in a code that the cell can read. RNA replicase is one of the enzymes that RNA viruses use, and it makes RNA from a template of RNA. And then you also have reverse transcriptase, which is used by retroviruses like HIV. So it basically makes RNA into DNA, making what's known as complementary DNA or cDNA, and then it basically takes off the RNA from this copy and it creates the complement to that DNA. So it starts as RNA, it makes cDNA, and then it makes double-stranded DNA, which is dsDNA. Once it's there, then it enters your genome, so that's when it goes into your DNA and it becomes a part of it. This is kind of showing you the same thing. So you have that positive single-stranded RNA, it makes a double-stranded RNA intermediate, and then eventually starts making copies of some of that single-stranded RNA. And this is, by the way, the way that you make the RNA genome. Here we have the action of reverse transcriptase. So the yellow section, that's the RNA that comes in the virus. So it uses reverse transcriptase to make cDNA. So this one right here, that's what it's doing. So it uses this template to make the red strand. And then the reverse transcriptase comes around again. It uses the cDNA that was made here, and then it starts taking off the yellow part, which is the RNA, and now makes a copy of double-stranded DNA. So double-stranded DNA is the same thing that you have in your nucleus. So once this strand is ready, it's going to go to your nucleus, and then it's going to become part of your DNA. So now that strand that came from the virus is part of your immune system cells. And every time that this cell replicates, it's going to have that little copy in there. So every new cell that is made has a copy of the virus. So how are they assembled? This process is not very well understood yet, but there are some enzymes that are involved in the cutting and the assembly of a lot of these viruses, because if they're not mature, they typically cannot infect cells. So typically, there has to be some other type of protein that helps with assembly. Once they're assembled, these virions end up exiting the cell. The first way, of course, is through bursting the cell or lysis, and the second one is budding. So budding happens like this. So it takes a little piece of the membrane with it, so that eventually it can trick another cell into letting it in. And the other is there's so many little virions inside of the cell that it bursts and they're basically just released after it explodes. So how are these transmitted to a new host? This replicative cycle, again, makes a ton of them. And the success of every virus has to do with its ability to make it through the environment from one organism to another. Again, the flu, for example, or the coronavirus, they're very transmittable because if you sneeze or if you're talking or anything like that that involves you opening your mouth, every time you do that, there's a lot of little particles that come out of your mouth. And if they land on somebody else's mouth or in their nose or somewhere where they can ingest it, it could potentially start to infect the other person. So natural selection favors viral alleles that allow the virus to replicate within the host and be transmitted to the new host. So again, natural selection typically favors things that aren't necessarily always going to kill the host, because if the host is dead, that means that it's not going to replicate to anybody else. So a lot of these viruses typically take a little bit longer to cause some of the symptoms, and again, during this period, they become infectious, spreading to a lot of people around them. So how do these viruses coexist with the host cell? The process of lysogeny is that process that I've already been mentioning. So the DNA that the virus makes becomes incorporated into your host cells. In the case of HIV, it becomes a part of those white blood cells. And during this period of lysogeny, there's no virus or no virions being made. It's just replicating your cells like it does normally. However, it comes to a point where then it begins to start working, and that's when you see the increase in the virus. Again, here we see that process. So you have infection, integration, replication, and then again, it can go two ways. You can divide cells that are infected, or you can go into the process of making virions. This dormant state is known as latency. So HIV does have a period of latency, anywhere between 6 weeks to a couple of months, even years. In terms of diversification, viruses don't have any machinery that proofreads their DNA because that DNA isn't really from a cell. So um, mutations tend to happen quite frequently in viruses. Uh, these polymerases that help copy the viruses, they're not as accurate as eukaryotic, which make like 1 in a billion nitrogenous base pair errors. These guys are a lot more prone to those. So because you have so many mutations, you typically see a lot of different strains of the virus or a certain type of virus. Um, where do viruses come from? Nobody really knows. There's three theories that have been hypothesized and considered by the scientific community. The first is plasmids and transposable elements. So maybe some of those elements that bacteria picked up from the environment or from other cells, maybe those would be the ones that ended up becoming, I don't want to say self-aware, but uh, able to replicate inside of a host. Then you also have the origin of symbiotic bacteria and at the origin of life. So again, here for transposable and plasmids, basically they consider them escape gene sets where they kind of ended up encoding some information, including the information to make a capsid, and then from there they were able to infect cells and get them to replicate them. In the case of the symbiotic bacteria, these bacteria that they were talking about, they hypothesized that they become degraded into viruses. So they changed the structure of their cell wall to be more like a modern virus. And then they had the loss of certain genes over time until they only kept the essential ones. And then at the origin of life is really interesting because nobody really, well, first of all, nobody was there, but it's also, we have so many questions still to answer. What if one of the things that was developing into the first cell had everything except for, cell membrane? What if instead of a cell membrane, it was encased in a protein shell, which could be a possibility? So what if that was the first go at life and it didn't work out, and then another one had a cell membrane with lipids? So then these two kind of started to grow, one of them of course infecting the other and replicating with it. So I mean there's a lot of different possibilities that we have, but those are the three that are currently considered. In terms of emerging viruses and diseases, HIV is one of the ones that has been around for such a long time and has caused a lot of death. So it is one of those that has probably jumped, and I guess that process is known as spillover. And influenza is another one that is the same way. Um, in the case of influenza, you have shots that are basically updated every year because there are little changes because of those high rates of mutation that basically make new strains just about every season. So they still have some similar characteristics that people used to target. However, they're not always the same. So it has 8 segments. So let's say that this is H1N1 and this is H3N3. So whenever these viruses go into the same cell, they can basically recombine into something different. So now this strain could be something like H1N3 or H3N1. So those combinations are what make this type of virus so dangerous, is that it's so easily modifiable and combinable that it makes it a nightmare to treat. So let's say that last year you got the H1N1 strain, right? And then this year, this is the one that's going around. So because your body was already exposed to the H1, which is probably, let's say that it's this purple one, once you get this infection and your body picks up that little projection, it's able to mount an immune response. But let's say that if, for example, you got a strain of H2N2, your body doesn't recognize, or your immune system doesn't recognize the H2 or the N2. Uh, so it's really hard for your body to fight that off. So that's why in these cases it becomes quite difficult to try to guess which strain is going to be more common. When it comes to classifying these viruses, they're not living things. However, we can establish phylogenies and relationships between them. Um, for example, the HIV virus can actually be classified, and it was traced back to an infection known as the SIV, which is the simian immunodeficiency virus. And this is that jump that I was talking about earlier. When it comes to interaction with animals, there's a likelihood that there's going to be a jump. So, for example, all the interactions with birds um, gave us the influenza virus. All the interaction with swine gave us the swine flu, and the interaction with these simians, um, in this case, gave us HIV. So now there are different strains of HIV. There's HIV-M, which is the main strain, which is the new strain, O is the outlier, and HIV-2, which suggests that there probably was more than one jump. When it comes to responding to a new virus, there's a lot of things that have to be taken into account. So you see a pattern here. So you have patients that have the same and unusual symptoms within the same place and within a short period of time. So the physicians who are treating these cases report these cases to any public health officials um, and organizations that are responsible for tracing these things like epidemics and pandemics. And they have to identify the agent and the origin of the outbreak, kind of looking for the patient zero. In 2002, for example, there was an outbreak of SARS, which is the severe acute respiratory syndrome. Um, and this is the same type of virus that the coronavirus that we have in the coronavirus or COVID-19 virus, which is the SARS-CoV-2 virus. It's the same class and family of viruses. But in this case, it popped up in southern China and spread to several countries. There were over 8,000 infections that were probable and 774 deaths, which was caused by a new coronavirus that was harbored inside of a horseshoe bat. And not, I just want to clarify that not every person in China eats bats. It's only a couple of people that practice that. Um, and again, it typically happens in places like wet markets, where they kill the animal there and then they give it to you so you can cook it at home, or sometimes they'll even prepare it there. But because of that interaction, that virus jumped, and that virus was actually also identified in a civet, which is a small mammal that is also sold um, in China. So probably that horseshoe bat transmitted it to one of those small mammals, and then those small mammals that were being prepared or eaten maybe got some blood on someone, they didn't wash their hands, and it made the jump. So in this case, we have a lot of different things at play. However, the outcome is always the same: is you have some type of animal interaction that leads to a jump into humans, and that's when it becomes really dangerous. And one of the reasons these are so dangerous is our immune systems are selected for. If you think about really early on in history, there's always two pathways for um, an infection to go. You're either going to die from the infection or you're going to survive. And those people that die, sometimes they don't get a chance to make babies. So those genes are selected against, and the ones that are favored are those of organisms with strong immune systems. So we're used to viruses. We have immune systems that fight viruses. However, when something new pops up, like this virus, for example, or the COVID-19 disease that's caused by that SARS-2 virus, these are something that our immune systems may not be used to in terms of the evolutionary history that we have, so they could be potentially lethal. The Zika virus is also a virus that had spread through South Central America, the Caribbean, and parts of Florida. In 2013, it began in Brazil, and they saw a lot of different cases there, as well as it spread to 70 other countries. In this case, this virus led to different conditions in the mothers, but also microcephalia or microcephaly in the babies that were born from there. Of course, in this case, it was treated with by eliminating the mosquitoes to try to prevent that. It's still an ongoing problem. It's not something that's gone away, and they are working on different types of treatment for it. When it comes to the lineages of viruses, they are classified using the Baltimore classification system, and it's based on the genomorphology, the nature of the host, and how it replicates. So your DNA viruses are class 1 and class 2, and these are either double-stranded or single-stranded DNA that end up going directly into the manufacturing of messenger RNA. Then you have your RNA virus, which begin as RNA. Some of them create double-stranded RNA. Some of them are already positive sense. Some are negative sense that have to make the positive sense. And then they can go into making RNA. So the whole DNA to RNA to proteins is kind of out the window now. Um, and of course, we have the reverse transcribing viruses over here that are some of the most intricate when it comes to their cycles. Here we have just a table that's showing you some of the morphology, the host, and the cycle of some of these. In the case of double-stranded viruses, we have things like smallpox and HPV, the human papillomavirus, as well as the herpes virus and adenoviruses. Um, so again, a while ago, HPV was one of those things that just popped up because people were getting, or women were getting cervical cancer. So they discovered that it was a virus that was doing that and that males were carriers despite not showing any symptoms. Then we have our single-stranded DNA viruses, so things like the parvovirus and the desmoviruses. In case of double-stranded RNA viruses, we have a lot of plant diseases, as well as the reovirus and the rotavirus, which are some of the ones that cause diarrhea in children, especially in places like daycare. And then we have our positive sense single-stranded RNA, which are, again, a lot of them are for plants, but we also have the rhinovirus, which is the one that's responsible for the common cold, polio, SARS coronaviruses, West Nile, hepatitis A, C, and E, as well as Zika, dengue, and yellow fever. So this is a very common. Then we have the negative sense single-stranded RNA, which are the flu, for example, measles, and Ebola. Then in the reverse transcribing, those are positive sense single-stranded RNA viruses. You have the sarcoma virus in chickens, other viruses in small mammals, as well as HIV. And then we have the double-stranded RNA reverse transcribing viruses like hepatitis B.